Then there was Louis Pasteur. He was the one who used the Swan flasks, or S flasks. They kind of looked like this. He tried to test whether or not basically nutrient broth could grow bacteria on its own. And he discovered that it wasn't just purely air that was the issue. It was actually like dust in the air that was the issue. More stuff on biogenesis. Should have that word somewhere. Should know all about cell theory and the remaining scientists who contributed to it. So the Jensens made the first microscope. Hooke noticed cells in cork. Van Leeuwenhoek saw bacteria in animalcules, saw little things dancing under the microscope, called them animals, basically. Brown found the nucleus. Schwann and Sleeden, one worked with plants, one with animals. They both recognized that, hey, everything that's living has cells, and they all have nucleus. Virchow basically came up with the idea that life creates life. Cells make other cells. And more or less, that was kind of like our first main day of the lesson. So hopefully you have all of those scientists and all of those terms down. Okay. The next day we talked about microscopes. So I hope you have a bunch of microscope type terms down. You should know light and electron microscopes. And hopefully you had these known for your test the other day. I will test you on the 4 different kinds of electron microscopes. So transmission electron microscopes, you put electrons through a specimen, but it gives you 2D images. Scanning electron microscope gives you the outside edge of a specimen, and it's a 3D image. Confocal, in my mind, I kind of think of confocal as being similar to the transmission. It gives you a bunch of 2D slices, but if you put all these 2D slices together, it makes a 3D image. And then scanning tunneling is probably the smallest one. It can trace the surface up to 2 million times zoom, which is just amazing in terms of how small it is. How much zoom does my microscope have in this lab?
No.
Yeah, I think the best one I have is either 400 or 1,000. 40 is the biggest ocular lens, but don't forget you're going to times the... Sorry, ocular is 10, objective is 40. So if you times them together, you either have 400 or possibly 10 times 100 for 1,000. This is 2 million. 1,000, 2 million, right? Like it's not even the same level, right? And so that's why I had all these pictures here showing you like the sorts of images you can get with electron microscopes. Like it's pretty crazy. So, you should also have these terms down. Staining. Sometimes you might need to stain a cell in order to actually be able to see the inside portions. Do you guys remember what we used for staining? Methylene blue, yeah. That'll work. And then centrifuge, basically it takes cells and it spins them around in a circle, and it should separate based on components due to densities. If you ever watch CSI, they actually use centrifuges in CSI, where like the guy will... Can you leave the console, by the way? Thanks. Anybody ever seen like CSI where they take like the sample, you know, the guy does a bunch of chemistry stuff in it, and he puts it like in the thing, presses the button, and starts spinning around in circles? Ever seen that? That's a centrifuge. Okay, third day, we talked all about cells, and this is probably where you've got to have at least a half page of terms on this. Organelles are the little parts inside a cell. Eukaryotes and prokaryotes are basically cells that have nucleus or don't have a nucleus, like say bacteria and viruses. Here are just like a million different terms you should know about a cell. Hopefully by now you know what they all are, like the basics of them. So membranes, nucleus, cytoplasm, wall, chloroplast, vacuoles, blah, 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 blah. Hopefully you have all those down, okay? At some point, you probably have already done this in terms of one of the questions on your assignment or the construction project. So I mentioned yesterday to some kids, if you're really smart, double up on the work. The question you do for number 5 on your assignment, which talks about all these different parts, copy and paste that literally and use it for your construction project. Right, like do it on the computer, just do it twice. Wow. Don't do it twice, do it once, and just hand it in twice. Does that make sense? Just be smart. What else do we got here? You should know about the phospholipid bilayer, which is what makes up the cell membrane. You should know the terms hydrophilic and hydrophobic. I remember phobic because phobias are things you're afraid of. So phobic things don't like water. You with me? No, you're not. Alright. You should know the term fluid mosaic model. That was on your test, actually. Hopefully you knew that one beforehand. You should know like semi-permeable, what that means. And then here was a whole list of words where when I wasn't here one class, I had you guys research all these and we talked about them the next day. So Brownian motion is basically the concept where particles are always moving around us. I like this slide here. Here's a particle, and it's slowly but surely going to move because there's like a million different collisions hitting it, and eventually it's going to work its way around the room. Does that make sense? Did I give you guys the analogy of me being in Boston for the Patriots game and getting carried along by the crowds? Okay. Like I like that analogy of like I'm getting swept along by the crowds because there's 1,000 people all trying to leave Gillette Stadium, and I'm kind of stuck with them, so I kind of got to move along with them. I think of that as Brownian motion. And then once you leave Gillette Stadium, concentration gradient means that here's everybody in the stadium, and slowly but surely they all spread out. So some people go north, some go south, some take the bus, some have the subway, some take their own cars. Does that make sense? And you try to spread out again. You should know about diffusion. You should know about how diffusion is one type of passive transport, but there are two other types. So there's diffusion, there is osmosis, there is facilitated diffusion, and within those there's some other random terms you should know about. There is the hyper, hypo, and isotonic. Long story short, if you put a cell into a hypotonic solution, what that means is the solution itself has less solutes, meaning it has lots of water. The cell will absorb all of that water and swell up. Does that make sense? It's actually what happens when you go in a bath. You get all wrinkly. The facilitated diffusion, you should know about the two different types of proteins. There's the transport proteins. Actually, transport and carrier should really be the same thing here. Did I use the word channel protein somewhere else at some point, perhaps? Yeah, you might actually use channel and carrier proteins. Yeah, here we go. Here's the word channel. Transport proteins could be channel proteins. Same thing. I like the analogy of like an airlock system for the carrier protein. I think I talked about this with a lot of you guys, the idea about how, like, say Walmart has like double doors when you go in. First you go in the first set of doors, then those doors close behind you, then you go in the second set of doors. In my mind, that's what I think of when I think of it as a carrier protein. Whereas a protein channel, you just walk through an open door, right? Like someone just walked through that door, that's a protein channel. Okay. Active transport, I'm sure I have on the next slide here. Active transport, you have to move things against the natural flow of concentration. Basically, that's me going out of the classroom and pulling some kid into the classroom and saying, get to class. Right? I use energy for that. Then there's also endo and exocytosis. These are when molecules are so big, you literally have to like absorb them into the cell. This is the analogy I have where I have a bed that I can't even fit through my doors. So we had to like take the window out and put the bed in through the window. You should know how to calculate a surface area to volume ratio, by the way. Even though that's not really a term on our list, you should know how to do the math at least. Okay. Last day that we had notes was the Magic School Bus Day. And I know I didn't actually go through the notes themselves, but make sure you have all of these key terms written down. So you should know the terms tissues as well as organs and systems. Even if I didn't actually highlight the word organ, that's a word you should know. Same thing with systems. For example, on a plant, an example of an organ would be a leaf. Right? Like a leaf is a type of organ. Or the stem would be an organ. Or the roots would be an organ. Or like the buds of a flower that are part of reproduction is an organ. Does that make sense? Meristems are the things that grow. We're actually going to talk a little bit more about that today, so don't forget that word. There's the root and shoot system, basically above and below ground. The actual plant leaf itself has a lot of different terms you should know. Cuticle is the top layer. It's transparent. Needs to be transparent so sun can go through it. The surface cells are called dermal tissue. Same thing. They're see-through. Chloroplasts are found in the next layer. This is called palisade tissue. Below that is the spongy tissue. And you see how this stuff's all thickly, tightly bunched together, and this stuff is not? The bottom half of the cell needs to allow gases to go in and out of it. The top half of the cell needs sunlight. Sun goes through here. Carbon dioxide and water go through here. Also within there, there's xylem and phloem. We have a name for that. We call those vascular tissues. It's kind of like your blood system, arteries and veins. Stomata, you should hopefully have down. Stomata are made of 2 guard cells. Normally those guard cells will be flat together, but if they fill with water, because there's enough water to actually undergo a photosynthesis, the guard cells will swell, creating an opening, and that opening can let through carbon dioxide so you can actually do your photosynthesis. I think I've talked about most of those words at some point here. Oh, sieve tube cells. I think I have it bolded somewhere else, but if you didn't have sieve tube cells written down, these are the cells that are alive but lose their nucleus. They carry the sugar in them. The companion cells are the ones next to them. I like the analogy of the big muscle guy, but he's kind of dumb. And then like the little short guy that's the brains of the operation. Like, just such a classic cartoon kind of concept, right? That's what I think of here. These guys don't have a brain. These guys are the brains. Does that make sense? But the phloem actually travels through the sieve tube cells. The important part is that they don't have a nucleus, so there's more room in the cell for sugar. Otherwise, there's just not enough room. More pictures with definitions of stuff you should know. More pictures. Guard cells I mentioned. You should know about photosynthesis. So hopefully you have that written down. You know the basic equation. Turgor pressure. I don't know that I mentioned that one when we did the Magic School Bus very well, but Turgor pressure is the name of the pressure that causes the guard cells to swell open. That's the name we have for the pressure that causes these guys to be tight together, and then you have that stomata opening. Transpiration is basically a fancy word for evaporation in a plant. And the reason why it's so important is that that's what helps draw water up the plant. That's one of 2 things that does it. Water gets pulled up the plant due to evaporation, and it also gets pushed up the plant due to something called root pressure, which I hope I have somewhere in here. I have root pressure in here. Here's transpiration pull. Did we talk about root pressure at some point? Yeah. That's a term you guys know. Apparently I just don't have it in the notes here. So make sure you add that. If I didn't talk about root pressure, basically the idea is that as more and more of the water particles are absorbed by the roots, they push, and the water particles also get pulled by evaporation. That's how you're able to get water to go uphill against the flow of gravity. Xylem, phloem, you should know those things there. There was a word somewhere here, tracheids. There it is. This is what xylem is made out of. They are dead cells. Tracheids don't have anything left other than just a cell wall. And that's what xylem is made out of. I think this is actually labeled wrong. I feel like this one's supposed to be xylem, and this one right here is supposed to be phloem. Because phloem are supposed to have sieve tube cells and companion cells, so I feel like this diagram is actually wrong. What else do we got here? Well, that basically covers it. Any questions so far? Okay, let's talk through our last set of notes then. So I've got a bunch more words to throw at you, so write them down as we go along, or just do it later. Before I get to that, I want to compare plants and humans again. I mean, both are living things. Both have cells. We all have nuclei in our cells. We reproduce. Well, I mean, albeit very different ways, right? But plants and humans are very similar. I want to talk about some of the things that the human body can do that are called stimuli. You guys ever heard that word before, stimuli? Yeah. Stimulus? Anybody ever been to the doctor's office and they do that thing where they like hit your knee and your knee shoots out like that? You know, you ever seen that thing before? I forget what it's called. There's a fancy name for it, but or anybody ever like had a doctor look at your eyes for a concussion and like they look at your pupils and see whether they dilate? Anybody ever seen or heard that before? Yeah. And like if your pupils don't dilate, that's a problem because you're not reacting to the stimuli. Right? Human body has various different things that it does naturally on its own I don't cause my pupils to dilate. I don't actually make my heart beat. That's another good example, where like my heart beats just on its own. You guys ever heard the terms like voluntary and involuntary?
Yeah.
Right? You can almost kind of bridge that into this here, where there are certain things that as humans, if I get knocked unconscious, I still breathe, right? Because the body takes over and causes me to start breathing on my own, even though my brain is kind of shut off because I'm unconscious. Does that make sense? Similarly, if I hit my knee like this, do I force my knee to do that? Well, no. If I hit it, that stimuli causes it to act naturally. Today I want to talk about a few of the stimuli that plants have that we've learned about. And these aren't like an exhaustive list, but there's just a few things we've learned about plants. The word we use is, we call it a tropism. Tropisms are basically like what happens after you apply a stimuli. So for example, after I hit my knee, what happens? Well, it kicks out, right? That would be the tropism. The stimuli is me hitting the knee though. Okay. So the first one is based on phototropism. And whenever you have like a tropism in a plant, there's always two halves to it. There's a positive and a negative. Okay. I mean, you guys know those words, right? Positive and negative.
Yeah.
Plants have positive phototropism on the top side of the plant, meaning they always grow towards light. But I gotta be more specific. They also grow away from the light, depending on which part of the plant you talk about. So you guys remember the terms root and shoot systems?
Yeah.
The shoot system grows, if there's like a sun up here, plants always grow towards the sun. The top half of the plant grows towards the sun. The shoot system grows towards the sun. But the bottom half of the plant, the root system, actually grows away from the sun. We call that positive and negative phototropism. Let me show you some more pictures here. Here's a picture of just some sprouts that are growing in a jar. You see how they're all growing diagonally? The reason why they're growing diagonally is that the sun must be somewhere over in this general vicinity. They actually turn towards the sun. There's actually a flower that's a very famous flower. It's actually named because of this. That specifically grows towards the sun. Anybody know what I'm referring to?
Sunflower.
Yeah, sunflowers. Yeah, sunflowers will always turn the heads of the sunflower towards the sun. Like they actively seek out the light from the sun for their photosynthesis. So, one of the cool things that you can do in an experiment, though, I'm going to skip ahead a slide here. Grow a plant sideways. I know that seems kind of ridiculous, but here's what would happen if you put the plant sideways. The stem and the leaves of the plant would eventually start growing towards the sun. Even though it's sideways, they'd actually start taking off in that direction.
Just like when you see trees growing outside of the middle.
Right. Like the tree branch starts going this way, but it eventually corrects itself. Yeah. But here's the weird part. The roots actually will start growing the opposite direction. Like the roots don't just kind of go down like this or out that way. Like they will very specifically go away from the sun. It's very, very unusual. Well, maybe not unusual. Like the top half of the plant goes towards the sun. Therefore, the bottom half of the plant has to go away from the sun. Does that make sense? So we call that phototropism. So I think I have the notes for that here. There's actually another type of tropism as well. We call it gravitropism. And plants will often also respond to gravity in terms of how they grow. Does that make sense? So that's kind of what this picture shows here, is that the leaves, they're eventually starting to grow up, and they're starting to grow down. That looks like it's evidence of gravitropism, where it's growing towards... Does that make sense? Oh, I got a picture there. It does a better job than I can. Okay, well, that's actually all I need to know for the tropisms. But I want to talk about two more scientists to add to our list. Darwin and his son, they recognized these sorts of stimuli, and they wanted to really focus on the phototropism, the whole idea that plants grow towards the light. So they did an experiment, and I've got a lot of pictures here. So I think I might just skip reading the section. You guys can read it yourselves. I'll just talk about the diagrams I have. Okay. Here's what they did in experiment 1. They just grew a plant. And the plant, naturally, like it does, started tilting towards the light. Which kind of makes sense. Plants grow towards the light. But that was called the control variable. You guys know those words, control, manipulated, responding?
Yeah.
Right, like this is the thing where he made it the same. Then he manipulated, and what he did is he put a little like covering over top of the tip of the plant. And he discovered that if he did that, the plant didn't actually bend anymore. And just to confirm that, he did kind of the opposite. Rather than putting a covering over the tip of the plant, he put a covering around the rest of the plant, but left the tip of the plant exposed. And oddly enough, in the third example, the plant actually started bending again. What does he infer from that? The tip is the important part of the plant then, right? The tip is the part that actually recognizes and responds to light. The actual stalk of the plant apparently is not as important as the tip. Okay. Then another scientist I want you to know about, Peter Boysen-Jensen, did another experiment where he wanted to focus more on, well, why does the tip of the plant actually matter so much? And so here was his basic experiment. He cut the very, very top of the plant off, he cleaved it right in half. Left the roots in the ground though, okay, just cleaved the top half off. And in one of the experiments, he put in between the tip, he tried to reattach it almost. So I cut it off and I go, oops, I'll just put it back and hope that it regrows. But in between there, he put a jello kind of compound. And the jello is permeable. And then on another experiment, he put a rock in between there. The rock is called mica, and basically the rock is not permeable. Nothing can get through there. And what he discovered, and I think this kind of makes sense, is that what was happening is that messages were being sent still through the plant that had a permeable cut through the middle of it. But as soon as he cut through the plant and had a non-permeable one, even though the tip of the plant was still there, it wasn't able to communicate anymore. Does that make sense? It'd be like, I don't know, cutting off of your arm and then expecting you still to be able to move your arm. Well, your arm was just cut off, right? Why would it still be able to move? Does that make sense? But in this example here, he reattached the arm and it was able to kind of keep growing. Does that make sense? Where in this example right here, the arm was cut off. He put a piece of rock in between there. It didn't keep growing. Does that make sense? So here's his conclusion. He concluded there must be a chemical involved. And he eventually isolated that chemical and had a name for it, which I have coming up sometime on the next slide. Here's another example of pictures, though, just to illustrate all these experiments. Here's the Darwin's experiment. So first experiment, light hits it. Oh, by the way, one thing I forgot to mention. Earlier in the lesson, we talked about something that controls the growth of plants. There's a name for it. You guys recall that name? It begins with an M. Meristems. Good job. Meristems. Yeah. What happens with cells in the meristem section is in order for things to grow, some sections will grow just a little bit faster than others. So for example, as a plant grows, if this one and this one, if they all grow at the same rate, this one grows and that one grows and this one grows and that one grows, the plant grows kind of steady. But what can happen with a tropism is this side can grow to there, but this side will grow higher, and then this side will grow higher and higher. And what ends up happening is you have cells over here that are say normal size cells, but there's cells over here. See how these ones are bigger, they've grown faster, and that allows the plant to start bending. Okay? The cells that are away from the light will actually be bigger, causing them to bend more. Let me say that one more time. The cells that are furthest from the light will actually grow faster because they'll grow bigger, causing them to grow more to allow the plant to bend. So these cells right here that are actually closest to the light are actually smaller. Seems kind of backwards, doesn't it? Yeah. Here's their experiments then. First experiment does nothing. Plant grows towards the light like you'd expect. Second experiment cuts the top off. Doesn't grow anymore, obviously. Third experiment puts just a covering over the top of the top. Plant's not dead, but it doesn't bend towards the light. Because it's like cutting the head off of somebody, right? It's covered up. Maybe not cutting off. Puts another tip on top. This time it's transparent. Well, now sunlight can make it through the tip, and now the plant bends again. Next experiment covers the base. Well, that doesn't actually seem to do anything. So they're able to infer that, yeah, it's the very, very tip of the plant that seems to recognize the light. Well, then Boysen-Jensen says, well, let's actually just cut the head right off and see what happens when I try to reattach it. Well, in this case here, jello seems to allow the movement of particles in between. But a rock, it stops. There's a barrier in between.
But it still grows, right?
Well, I actually don't know that it still grew, actually. Do I have that in my notes here? It might have still grown. I need to research this. The more important point was that it didn't bend towards the light, though. I'll look into that, Gabe. That's a great question. Did I say that? I think all I said is that phototropism did not occur. Yeah. I'll look into it. Okay, last thing. Last scientist I need you to know. F.W. Wendt. His goal then was to try to discover, well, what is it that causes this? And he eventually found a hormone. You guys have maybe heard that word before. Like, humans also have hormones in their bodies, right? Different chemicals that stimulate growth. And the chemical that he named then called it auxin. And basically the concept is that here's the sun. These guys over here, the ones that grow more, they have more auxin. So, just like in the same way that, like, say, there's testosterone and estrogen as, like, hormones that boys and girls have. Well, this is the hormone a plant has. So there's more auxin on the side that grows more. It grows more so that it can bend towards the light. Does that make sense? Auxin. Cool. Okay, I'm done. That's my last slide, right? Okay. Quick summary, then you guys can scatter if you want. Here's the plan going forward. Give me 1 minute, guys. Then you guys can scatter. Okay. One of my goals is to, I want to challenge you, but I want to give you a bunch of things to work on at once. I think that's actually an important skill. I don't want to say just work on this, then work on this, then work on this. I want to give you 5 tasks and just give you a lot of time and say work on them all. So we're actually done our unit, believe it or not. Bio is a very different kind of science than chem and physics. It's not a process-based one. It's very much a remember and regurgitate type of science. So if you haven't made a fairly substantial word list to look over, you should. You should always read your textbook, by the way. Read over your notes, because there's a lot of memorization. Okay. In terms of going forward, then, we have, I think, 7 remaining tasks. And some of these you might be done already. So the first 2 tasks, finish off the lab activities we did. I know a lot of you have them done. So that's 2 tasks. Then 3 more tasks. I'm guessing that most of you are going to choose to omit one of those 3. But you should work on the research paper, the construction project, and the multimedia project. If you're smart, though, you probably can reuse a lot of the information from those projects as well as the assignment. And then the only other things we have to do, Josh.
What?
Hi. I'm kind of talking to people. You're one of the people. Okay. The last thing then, next week, we're going to do a quiz on Tuesday and a unit test on Thursday. Any questions? Okay. You have a lot of time. You have rest of today, all of Friday, all of Monday, all of Wednesday. Scatter as you need to, but use it well, okay? So let me know how I can help otherwise. I'm done.